So our next speaker is a dear friend of mine. She's smiling, which means she knows I'm going to ask her for something. Dr. Cheryl London. Dr. London is a veterinary medical oncologist with over 20 years of experience in the field of translational and comparative oncology and therapeutics development. Her work uses spontaneous models of cancer in dogs to answer key questions regarding the elucidation of targets for therapy and then to translate these findings into pre- and post-IND settings. As the director of the Clinical Trials Office at the College of Veterinary Medicine at OSU, she's responsible for overseeing all the studies in client-owned animals, that means your pets, with spontaneous disease. Dr. London also directs veterinary clinical research shared resource of the OSU Comprehensive Cancer Center and is the director of the same thing in the Center for Clinical and Translational Sciences. She really started a new program, the CCTS, now probably almost 8 years ago, which is a translational therapeutic think tank, which is responsible for coordinating translational studies in the pre-IND realm across the OSU colleges and programs, and bringing multiple areas of expertise together to actually improve study design and conduct. Her broad research experience in signal transduction pathway biology has been successful in identifying novel targets for therapeutic intervention for cancer, such as osteosarcoma. She recently moved her research laboratory to the Molecular Research Institute at Tufts and now holds dual appointments together at Ohio State University and Tufts University with the goal of creating strategic collaborative alliances to further comparative and translational oncology. Cheryl, it's wonderful to have you with us today, and she's going to talk about translational research. Wherever you are, behind me.
I'm relatively small, so it's easy for me to disappear. So most of everything that you've heard about today really revolves around the world of human medicine, but I'm going to talk a little bit today about what I do, which is translational medicine, and focus in the realm of translational oncology because that's where we've made most progress. But I will give some examples where we're making differences in non-oncologic diseases. So I'm a veterinary medical oncologist, which means that I treat animals with cancer, but for a long time I've been involved in the world of translational oncology and comparative oncology, where we try to leverage dog cancers to study new drugs and learn new things that may help the human drug development process. And so why do we do this? We do this because the failure rate in human oncology drug development is abysmal. And actually, the failure rate in drug development overall is abysmal. And if you look at likelihood of approval of most drugs, it's fairly low. For many of them, it's between 10 to 15% from start to finish. But if you just look at the oncology drug likelihood of approval, it's sitting around 7%. So 7% of drugs that started from phase 1 actually end up being approved. Which is really not financially sustainable in the long-term setting. It used to be that for $1 billion of money invested, you could get between 40 to 50 drugs approved. Now it takes more than $1 billion of investment to get a drug approved. And you can imagine how much money is wasted when you make it all the way through a phase 3 study, $1 billion in, and your drug's not approved because it didn't work, or it had a side effect that wasn't acceptable. And the other day, I was sort of rooting around CNN, reading all the bad news, and then actually found something that I thought might be good news, but turned out to be bad news as well, which was sort of a perspective on where we're at in cancer drug approval. And so there really has been a flurry of new drugs approved for treating cancer on the human side. But when you look at the impact that has had in patients overall, it's actually pretty low or pretty minimal. So if you look from 2002 to 2014, there have been 72 new drugs approved to treat cancer. But if you look at the overall impact that has had on patients, an average of 2.1 more months of life. So we hear a lot in the news about new cancer therapies, and some of them really have made a difference. There's no question that immunotherapeutics have made a giant dent in some cancers. But I think we get sort of overwhelmed by that positivity and miss the fact that even with those positive results, there are many drugs that are approved that are having little impact on patient outcomes. So why do these drugs fail? The overwhelming reason is efficacy. So they get through a phase 3 study and they just are found to not be effective. Or there's a safety flag that arises later on in the course of study. So you have to ask yourself, how can you spend $1 billion and get to the point where you just didn't know that your drug wasn't going to be effective, or you didn't know that it wasn't going to be safe? Like, that just is astronomical to me, that you spend that much money and don't know that it doesn't work and that it has a terrible side effect. So why is that? So I would argue that to some degree, the reason why we fail so miserably is because we rely so much, so heavily, on models that really don't recapitulate disease. And in the case of cancer research, it's the mouse. So we use mice for many, many, many disease models. And we have found over time, even though we try to ignore it, that much of the data that we generate with respect to clinical outcomes in mice doesn't mirror what happens in people. I don't know how many of you were aware of the sepsis story in mice, that for years mice were used as models for sepsis, and then when they finally did the profiling, molecular profiling of the sepsis response in mice and compared it to people, it was totally different. So that's why all the clinical trials that were based out of mouse data and moved into people failed, because the model didn't really recapitulate what was happening in people. So if you look at mouse models of cancer, many of them use immunodeficient mice that lack critical components of the microenvironment. And as we know now, and you heard in the last talk, the microenvironment is really critical in driving how tumor cells grow. And if you don't address the microenvironment, you're likely to fail. Cancer cells or cancer typically grows in mice in a very, very compressed timeline, so often in a period of 1 to 2 weeks, whereas cancer in people grows over months, if not years. So really compressed timeline, which changes the dynamics of those tumor cells in relationship to their microenvironment, which then changes how they respond to therapeutics. Mouse cancers are also relatively homogeneous when you compare them to human cancers, which are, incredibly heterogeneous. It fits in with the whole James, no two tumors are alike, everyone's cancer is different. And the other thing that I think is really important when we think about therapeutics moving forward into humans is that mice tolerate therapeutics far better than people do. What's our sort of endpoint with mice? How do we decide if they're not doing well? Well, they have ruffled fur or they die. So that's pretty much, or maybe they lose some weight. So, but how do you ask a mouse, is your quality of life impaired by taking this drug for 3 months? Like, you can't ask that question. And chronic toxicities in people associated with therapeutics, it's a huge problem, particularly if you're going to be on a therapeutic for a year and it makes you feel lousy. So you can't really assess that in mice. The other thing that I would argue plays a huge role in why we fail is our overestimation of clinical relevance of results. We often look for endpoints, either in vitro or in mouse models, that are not biologically relevant, although they may have a p-value next to them. We've become so reliant upon statistics that we ignore often the obvious. If you want a therapeutic to work, I don't care that there's a p-value there. I want that blue line to be down there. I don't want that tumor to grow at all. I don't want it to grow less fast than the control. But we take this data and we say, this drug was effective. It slowed the tumor growth down. Look, there's a p-value there. And so then that becomes part of this positive data set that moves a therapeutic along. And I call this the 4-letter word bias, which is a cumulative bias that we as scientists really try not to do, but inherently, in some ways, we're driven to do it because we have to form a hypothesis about what our drug might be doing. And then we want to generate a set of data that supports our hypothesis. So we always pick the figures that look the best, that show the best statistical endpoint, so that we can tell a story and publish a paper and then submit a grant and then get money to do more research. And even in the pharmaceutical side of things, in the industry side of things, if you have a molecule that you spent a lot of money on, that you've developed, and it has a growth curve that looks like what I just showed you, often that once the train leaves the station, it's very hard to get that to stop in the industry side because you've invested so much time and effort that even if the differences that you see are minimal, that drug keeps moving forward. So we have cumulative bias, we have overinterpretation of our findings. So how do we unbias the system? And I would argue that dogs with cancer unbias the system. In many ways, they can serve this purpose. Because unlike mice, they develop tumors spontaneously. They often occur over a long period of time, so tumors grow over months to years, and they exhibit a wide degree of heterogeneity, and I'll go over that. So cancer is one of the most common diagnoses that we see in the dog population. If you look at the number of diagnoses per year of neoplasia, it's over 4 million, which is huge, and that 1.5 million of those are malignant diagnoses. So huge population of dogs every year that get tumors. And these are spontaneous tumors. These are dogs that you and I own. If you look at the actual annual age-adjusted cancer incidence in dogs, it's higher than people, 381 per 100,000 compared to 300 per 100,000 in people. So dogs get a ton of cancer. And you look at the spread of cancers. They're a little bit different than some of the cancers that we see in people. So dogs tend not to get colon cancer. They don't eat McDonald's, and they don't drink. And they actually eat dog food that's very high in fiber, so that's probably why their gut is protected. But they get a lot of things like lymphoma. They do get some adenocarcinomas, lots of sarcomas. Many of the diseases, the cancers that we see in dogs are sort of mimic pediatric cancers, particularly osteosarcoma. And then if you look at cause of death in juvenile versus adult dogs, neoplasia really it takes the cake here. So cardiovascular events are way down here, and we just don't see heart attack and stroke in dogs, and dogs basically die of cancer. That's why most dogs die. And if you look at when cancer starts to occur in dogs at age 6, it starts to rise, it peaks at age 10, and then starts to drop off at age 12. So if you make it to 14, you're probably not going to get a tumor. So why should we bother studying cancer in dogs? Well, we know now that there's greater synchrony between dog-human genomes compared to rodent and human genomes. So if you think about genes and chromosomes, they're all the same cards in the deck of cards. They're just shuffled a little differently. When you look at how they're shuffled in people and dogs, it's closer than it is when you look at people and mice. So it's easier to find associations in dogs and apply them to humans than it is sometimes in mice and apply it to humans. We know that many of the signal transduction pathways that are altered in human cancers are similarly altered in dog cancers. And really, I think one of the most important things is that it really recapitulates the heterogeneity that's seen in human tumors. So we get recurrence of tumors, we get metastasis that's spontaneous, and we get resistance to therapy. So we use the same therapies that are seen are used in people. We use radiation, we use chemotherapy, we use surgery, we have immunotherapy, we have small molecule inhibitors. So the spectrum of occurrences in the human cancer world occur in dogs as well. It's really actually pretty cheap to do cancer clinical trials in dogs. If you look at the average cancer clinical trial that we do, it spans somewhere between $100,000 to $200,000. When you talk about cancer clinical trials on the human side, you're talking about millions of dollars. So much, much cheaper, and we don't actually need to use GMP product. We can use GMP-like product. Another really critical feature of dog cancers that enables us to add value to the human side is that we can treat our patients before they've been treated with any other therapy. One of the huge problems that's encountered in human clinical trials is that in order to enter a clinical trial, you often have to have failed standard of care. So most of the patients that enter trials for new drugs have multiply failed. They may have comorbidities, and they're a much more challenging population to treat. So a drug combination that may have worked in the original setting when the cancer was first diagnosed now isn't working in a patient that has diffusely metastatic disease. But how would you know that? So you could miss an opportunity for a drug to work well because you gave it in the wrong setting. So we can treat our patients with novel therapies before they've seen any other therapy. And then lastly, the body size of companion species. So we can multiply sample dogs. So we can take multiple blood samples, we can take multiple tumor biopsies, we can image them using the same modalities. You really just can't do that in mice effectively. You can't follow mice linearly the way you can in dogs. So it gives you a better sense of patient progression over time. The other thing that we have that's a big advantage is something called the Samespecies therapeutic index, which is that because we do study drugs and devices in laboratory dogs first, we know how those drugs and devices are going to behave in our client-owned dogs. So we have a very good sense of what the adverse event profile is going to be, what the PK is going to be, what the PD is going to be. So we know all these things before we actually get into our patient population. That Samespecies therapeutic index really facilitates our ability to move very quickly from research dogs to client-owned dogs and allows us to scale up or to dose escalate much more rapidly than they do on the human side. So if you were to think about how one could effectively integrate dogs with cancer into a drug development scheme, this is sort of what it would look like. You would take your preclinical models and still use them because they're important. Mouse models are critical for us to understand biology. You can move genes in and out of mice really easily, so it helps us to dissect pathways. But they're not great for really assessing whether a therapeutic is going to be successful in a person. So you still need to do that, but instead of going right into humans, take it into the dogs first and ask some really critical questions. What's the activity? Is there a toxicity that we didn't know about? What is the PK and PD? And once you have that established, and you haven't had any red flags, then you go ahead and move that into people. And simultaneously, while that's in early human studies, you go back to the dogs and ask questions that relate to what's the dose, what's the regimen. Regimens can make a huge difference in whether or not a drug works. What's the schedule for giving it? Are there biomarkers that we can use to predict response to therapy? What are the responding histologies, or what are the responding molecular profiles? And how can we combine this with other therapies? And that's another big challenge on the human side, doing combination therapies. How can we effectively screen which combination therapies are going to work well? That's relatively easy to do in dogs. And then if this data looks good, then you use that to inform your subsequent phase 2 and phase 3 studies. So what could this do? What it could do, what it's predicted to do, is to potentially remove a lot of drugs from the pipeline early on, so they never make it into those early phase human trials. And then you would have a lower cost due to this integrated drug path. So the blue line is the integrated path. So less drugs that get pushed through, less cost overall because you have less failure. So it could potentially save millions, if not billions of dollars. So I'm going to give you some examples where we've actually engaged in this process and facilitated the drug development in the cancer realm. And then I'll give you one last example where we were tasked by a company to do this for them, and then they didn't listen to us because they believed their mouse data, and then they failed their phase 3 study. And then they're no longer in existence. So I've actually worked on KIT as a target for therapy for many, many years. KIT's a receptor kinase that's critical for the development of many cell types, but particularly mast cells and melanocytes and others. It has a ligand called stem cell factor. It's anchored in the cell membrane. And many, many years ago, when I was in my residency, we identified the presence of activating mutations in KIT in a type of tumor called mast cell tumors in dogs. And this is actually the second most common tumor that we see in dogs. It's rare in people. So this is an instance where the dog tumor isn't a model of mast cell disease in people, it's a model of KIT mutation for people. Because we see so many dog mast cell tumors, and about 30% of them have activating mutations in KIT. And these activating mutations mirror activating mutations that are seen in human KIT and other cancers such as GIST. We knew that these mutations were associated with high risk of recurrence, metastasis, and were a driver of disease. So Dogs are a good model for KIT dysregulation because we see so much KIT mutation in the mast cell tumors. And they're a particularly good model for activating mutations that occur in 50% of human patients that have GIST. And with that knowledge, we were approached by a company called Colton that was interested in studying KIT as a target for immunotherapy. There are several small molecule inhibitors that are already out there that target KITs. How many of you have heard of Gleevec? So Gleevec is the first one that was approved, and that was approved for treating actually CML, but is also a KIT inhibitor and blocks KIT signaling in GIST. That was approved probably like a good 20 years ago, I think. And so we've had all these small molecule inhibitors that target KIT, but there's been a strong interest in using an immunotherapeutic an antibody-based therapy to inhibit KITs. So KTN-0158 is a humanized anti-KIT antibody that was being developed as a potential therapy for cancer and mast cell-related diseases. What was important about this is that while Colton was developing this antibody, Novartis had made an antibody that they had put into human clinical trials. It was actually an antibody drug conjugate. It had cleared its primate testing, non-human primate testing, and the first 3 patients that received that drug anaphylacted and almost died. It degranulated their mast cells, and that was not predicted based on the preclinical work they had done in rodents and primates. So there was huge concern on the part of Colton that they were developing an antibody that was going to fail up front. The FDA was concerned about putting more anti-KIT antibodies into people because of this anaphylaxis event. And in a fortuitous set of events, it turns out that the antibody bound canine, feline, non-human primate, and human KIT with very high affinity, but it did not bind rodent KIT, which sort of was the gateway for them coming to us and saying, we'd like to evaluate this antibody in dogs because we can't do it in mice. And so we knew that it was a potent inhibitor of wild type and some oncogenic KIT, and that it did modulate mast cell function and survival. So this is the way this antibody worked. It basically inhibited KIT dimerization by binding to this D4 region, which was a slightly different region than the Novartis antibody bound to. It bound up in here in the D2 region. It was a potent inhibitor of proliferation of KIT-driven cells. So this is the antibody inhibition compared to the small molecule inhibition. So it was much more potent. And then we were able to show that it was equivalent in its ability to inhibit KIT phosphorylation here, inhibiting KIT phosphorylation in dog KITs compared to Gleevec, which was equivalent really inhibiting KIT phosphorylation in dog KITs. So the first study that we did was a very small safety study in healthy research dogs, where we took them, gave them a single dose of antibody, and asked, does it deplete mast cells in their skin? And it turns out that dogs are a great model to study anaphylaxis. Their mast cells are super sensitive to anaphylaxis. They degranulate in response to chemicals. And so if you don't get a degranulation event in dogs, the likelihood is it won't happen in people as well. Because dogs are so sensitive. So we were a little worried that we were going to see anaphylaxis with the administration of the antibody, but we cleared that. So they didn't have any anaphylaxis, and we could show that within 7 days we had depleted mast cells in the skin. So it actually did have a biologic effect. The KIT antibody did. And you could see that at the higher dose of antibody, we actually held that depletion down for 28 days. So with our safety study, and again, this was 4 dogs. We just had to do a 4-dog safety study before we moved into client-owned dogs because we had Samespecies therapeutic index. We took dogs with measurable mast cell tumors, and we initially were going to look at 10 and 30 mg per kg doses, and ended up adding another schedule in. So that's the other great thing about veterinary clinical trials is that we're not regulated by the FDA. We don't have to have any FDA approval before doing these studies. So for us to change a regimen simply requires an amendment. And often this amendment gets processed in 2 weeks. So no red tape. So we can respond to what we're seeing in our patients really rapidly. So we studied these two schedules. We were hoping we were going to be able to give the antibody more than once. This is a humanized antibody. We knew that the dogs would make anti-human antibodies, but we thought we could get away with it in 21 days, and it turned out to not be the case. We did serial tumor biopsies and blood sampling and weekly assessments. So again, we can repeatedly biopsy these tumors, and we can take multiple blood samples. So we were actually really floored by the response that we saw. Across the board, we had almost every single patient have some shrinkage of their tumor. And this is based on RECIST criteria. And this is, for many of them, was a single dose of antibody. Some of them it was twice, some of them it was once. But you can see we had multiple patients that had shrinkage of their tumors, and some of those were KIT mutant dogs as well. Importantly, we were able to assess KIT phosphorylation within the tumor, and most of the dogs did target modulate, including the 2 dogs here that were evaluated and had KIT mutations. So we could show that we shrank the tumors and that we downregulated KIT phosphorylation inside the tumors based on the multiple biopsies. And this is an example of one of our patients who had failed probably 5 prior treatments, radiation, chemotherapy, surgery, and small molecule inhibitors, and another clinical trial. And here's her huge tumor, and within 2 weeks, the thing was markedly reduced. And if we had been able to continue to treat her with a canonized anti-KIT antibody, she probably would have continued to respond, but we were only able to give her 1 treatment because the dogs eventually developed neutralizing antibodies. So here's our response to therapy. And what's interesting is that many of these dogs went to surgery after the trial was done. And so we actually had full histopathology on their tumors. And because we were using RECIST to measure, which is the longest diameter, we thought there was still tumor there in many of those cases. And then when we looked at them histopathologically, it was just scar tissue. So here's an example of a dog that had a near partial response to therapy. And when we removed the tumor at the end of the trial, there was actually no tumor left there. So it was a histopathologic complete response to therapy. And then the other sort of interesting thing about some of these dogs is they had metastatic disease in the lymph nodes. Those were removed at the time of surgery as well, and there were no metastatic cells left. So our activity was much higher than we predicted based on our clinical impression, and it was much higher than we expected based on some of the preclinical work. But importantly, we had a really large safety profile, and we did not see anaphylaxis. So this is looking at the PK associated with the antibody. You can see that there's dose-dependent, dose-proportional PK. So the higher dose, you have higher persistence of antibody levels. But this was what the problem was, that in the dogs that we dosed twice, you can see that with the second dose, there was very little that was left in the bloodstream within 7 days. And they were able to detect within 10 to 12 days these anti-KTN-0158 antibodies. So we did develop neutralizing antibodies. So the moral of this story was that the dogs were able to be used to show that the drug was safe, that it was effective, and this was important data that went to the FDA. So this data was packaged into the IND, and when they went into the FDA, the FDA used this data to show that to understand that the drug was likely to be safe and unlikely to cause anaphylaxis in people. The other thing that was really important about this set of data was that the company was struggling with what disease entity to use this antibody, and they had a couple of different choices, and they ended up choosing GIST because of the correlation of KIT expression and KIT mutation status, and how comparable that was to what we see in dog mast cell tumors. So the human patients have been dosed. They've not had any anaphylactic episodes, and that phase 1 study is ongoing. The next story that I'm going to tell you is one where we were working with a company who was interested in targeting PI3 kinase as an anti-cancer therapeutic. So PI3 kinase is a critical pathway in all cells, but it's particularly important in cancer cells. It drives many functions of tumor cells, including survival, inhibition of apoptosis, metabolism, et cetera. In particular, PI3 kinase dysregulation is known to occur in lymphoid cells, B and T cells. And the one isoform that's typically exclusively expressed in lymphoid cells is PI3 kinase delta. which has become a target for therapeutic intervention in lymphomas. There are, unfortunately, a plethora of drugs out there now being tested that sort of intervene in the PI3 kinase signaling pathway in multiple places. Drugs that are straight isoform-specific PI3 kinase inhibitors or pan-PI3 kinase inhibitors and dual PI3 kinase mTOR inhibitors. So, I mean, you can understand why markets get really crowded because there's all these drugs are really targeting the same pathway. So we were approached by a company that had a novel PI3 kinase inhibitor that was primarily a delta inhibitor because they knew that they wanted to target just delta for two reasons. One, delta dysregulation is known to occur in both B and T cell lymphomas, and there had been some evidence that delta inhibition can correlate with response to therapy in human patients with diseases like lymphoma. But the other reason was delta signaling seems to drive survival of regulatory T cells. So there was a real interest in potentially inhibiting delta signaling to dampen the regulatory T cell response and then allow an immune response to occur in patients. So they approached us with this drug to ask whether or not we could do a phase 1 clinical trial in dogs with newly diagnosed or relapsed T and B cell lymphoma. And they had already done some safety testing in normal dogs, so we did a standard dose escalation study based on data that had been generated in the healthy dogs, and we did a bunch of pharmacokinetics and a bunch of pharmacodynamics. And we found that we actually had quite a bit of activity. So these are just waterfall plots of dogs that had response to therapy at multiple dose levels. However, we had at the daily dosing hepatotoxicity that hadn't initially been flagged. And we changed this to Monday through Friday dosing to allow them to wash out because when we did the interim pharmacokinetics, the drug is so orally bioavailable that they were drug accumulating over time and their blood levels were so high, way higher than we actually needed to hit target. So when we dropped to the Monday through Friday dosing to allow a washout, we markedly reduced our hepatotoxicity. And I should tell you that all of this sort of data, you know, we started enrolling dogs. We enrolled them pretty quickly because we see a lot of lymphoma patients. So we had most of this done within the first probably, I'd say, 7 to 8 weeks. And when we started seeing the hepatotoxicity, my suspicion was that we were drug accumulating. We ran interim pharmacokinetics, and the next day we changed the protocol. It was that easy for us to adjust what we were doing. And this is an example of one of our patients that had a T-cell lymphoma that was, there was a mediastinal mass and there was diffuse fluid in the chest, and this dog had failed other therapies, had a complete response to therapy on single-agent drug. And so this was our pharmacokinetic data, and you can see that we're in the micromolar range within, this is the first dose of drug, within the first dose of drug that this dog's ever seen, these dogs had ever seen, at 10 mg per kg, 3 micromolar. And then you can see with the higher doses we start to drug accumulate, particularly at the 25 mg per kg. This is our PD assay. We were able to show in dogs that had their first dose of drug, so they'd never seen drug before, we got a sample of their lymph node before drug and then 2 hours later. And you can see that AKT phosphorylation was downregulated in nearly all of them. So we had target modulation within 2 hours of the first administration of this drug. And what that tells you is that you only need actually this level of drug here, 3 micromolar, to inhibit target. So we were very rapidly able to draw a correlation between our pharmacokinetic doses needed and the pharmacodynamic response. So we knew that we didn't have to dose much higher than 10 mg per kg to inhibit target because all we really needed was 3 micromolar plasma levels of drug. So to summarize this vignette, the reason why this was important was that it first of all flagged hepatotoxicity for the company, which wasn't initially flagged as an event. And after we've worked on it a bit, it's turned out that the hepatotoxicity is due likely to inhibition of the regulatory T cell response and diffuse infiltration of the liver with activated T cells. So based on biopsies, it looks like we're getting modulation of the immune response, which was sort of surprising. And then in the human patients where they've seen this, they've now gone back and given them high-dose steroids, which seems to dampen down that infiltration of the liver with these activated T cells. We then went on and did a phase 2 study in dogs to confirm what we saw, and we had a 70% objective response rate, which was very, very high, and confirmed the dosing regimen. But the important part of all of this was that this data supported the subsequent application of their companion human drug in T cell lymphoma in people. They were trying to decide whether to head into B cell or T cell, and they opted for peripheral T cell lymphoma based on the fact that we had quite a few T cell lymphoma patients actually have complete responses to therapy. So the data that was generated in the dogs helped to guide where that drug was placed in people, and also helped to guide dealing with the adverse event profiles that they subsequently saw, which had to do with the hepatotoxicity. So the last story that I'll tell you is the one where the company didn't really want to listen to us, and their phase 3 study failed. So many years ago, I was approached to work with a company that was interested in heat shock protein 90 inhibitors. So heat shock proteins are critical for folding client proteins. They're required for cell survival during stress. They're basically named based on their molecular mass, and they're essentially chaperones, so they have clients that they fold. In cancer, HSP90 has emerged as one of the most critical players. It has a number of client proteins that are oncogenes, like KIT and others, and transcription factors. And we know that ATP is required for heat shock protein 90 to work. The other thing that we know is that in cancer cells, HSP90 has a lot of friends associated with it, so it's in this super chaperone complex that makes it very, very active. And the reason why it's so active is that tumor cells are dividing and growing really rapidly. They're making lots of proteins. Many of them are mutated, and they're hard to fold. So you have to have very high HSP90 activity. And this has a much higher affinity for ATP than the inactive complex in normal cells. So there's a differential targeting. So this company had developed a novel HSP90 inhibitor called STA9090 or ganetisib. And they had shown in vitro that it worked very well in inhibiting HSP90 and degraded a number of client proteins, worked very well in multiple mouse tumor models. For reasons that were not clear to me, they chose to move this drug forward, which is not water-soluble, had to be given in a very difficult diluent in order for it to be given IV. Yet they had this highly soluble prodrug that they chose not to move forward, and we ended up working with that because it was water-soluble and much easier to work with. And this is cleaved to the active compound in the plasma right away. So this was a much more facile drug to work with than 9090. And so they tasked us with looking at the biologic activity, essentially, of 9090 using the prodrug in a phase 1 study. And they had done some normal testing in dogs before we moved into our client-owned dogs, and we had 2 dosing regimens that we were going to test, 1 hour once per week and a 1 hour twice per week infusion, look at toxicities, establish biomarkers, and look for activity. And so, phase 1 studies can be really, really disheartening because often the drugs don't work. And so we had most of our patients fail to respond to therapy until we had one dog who was a crazy German Shepherd who was getting his infusion, and his catheter must have migrated out pretty early in the process, and all of it went subcutaneously, which gave me a little bit of a heart attack because I didn't know if the drug was a vesicant and his leg was going to fall off, but thankfully it wasn't. But he was the only one in the cohort that responded to therapy. And fortunately, we had captured his pharmacokinetics. So this was what his pharmacokinetics looked like compared to everybody else. So you give it IV, you have sort of this steep drop-off. If you give it sub-Q by accident, you have this long exposure in the plasma. So we went back to the pharmacologist and asked, how can we model this so that we don't give it sub-Q and can actually give it in a manner that is conducive to repeated consistent blood levels? And so we did a whole bunch of modeling and ended up saying we need an 8-hour infusion. And so we went and added a cohort in of dogs that got 8-hour infusions, and that's where we had all of our activity. So here's a dog with metastatic thyroid carcinoma. It's really hard to see this here, but had a really nice partial response to metastatic lesions. Metastatic osteo, had a really nice, durable response to therapy of the metastatic lesions lasted 5 months, which is pretty amazing because it's a really refractory disease. This dog went for almost 2 years. And that's where we saw all of our activity. So this is the PK on the 8-hour infusion. And then this was the PK with a 1 hour once or twice per week. And this was where we had all of our activity at the 8 hours once per week infusion. So we were able to tie the responses to therapy to blood levels of drug that were between 200 to 600 nanograms per mil for 8 to 10 hours. We were able to generate data that showed that you could show upregulation of HSP70 as a surrogate biomarker of HSP90 inhibition. And so what happened was we showed this data to the company, and they said, no, that's not what we see in mice. We just give mice an injection, and these tumors melt away. And we said, well, that's not what we're seeing in the dogs. And so the person who was in charge of preclinical modeling at the time just didn't believe the data. So he actually left the company, and a new person came in, and he went back and looked at the data and then did a few things. And he said, you're right, actually. When we went back and looked at the mouse data more critically, what happens is if you give a bolus of HSP90 inhibitor, you get a very acute downregulation of HSP90 function, but you get transcriptional upregulation of HSP90 and a bump back within 24 hours of protein expression. So you get an immediate compensation associated with inhibition of the activity. So if you look early, you'll see HSP90 inhibition. If you look 72 hours later, it's right back up. And so if they had bothered to go back initially and look at that time course, they would have seen that you get this bounce back effect. And that's why that short infusion wasn't really affecting patients, because you weren't really getting sufficient inhibition of HSP90. So then we went back to really show that this was the case in dogs. So we took the drug, 1474, and we gave it to dogs using 4 different protocols. And these dogs all had mast cell tumors. So in this case, we were going to use KIT as our client protein for HSP90 and follow KIT expression to see if we could show that giving that drug over 8 hours really suppressed the downregulation of HSP90 by showing that we lost KIT. The problem was that you cannot do an 8-hour infusion in people with 90/90 with ganetospib because it's not soluble in any normal substances known to man, and the limit on infusion is 3 hours. So they couldn't do an 8-hour infusion in people. So what we then asked is, can we find a protocol that of drug administration that mimics what we see with 8 hours? So we worked with the pharmacologist again, and we came up with this protocol, which is we're going to take dogs with mast cell tumors and give them the same dose equivalent drug. So they all got 6 mg per kg per week, and they got it 1 hour, over 1 hour once per week, which we thought wouldn't work, 8 hours once per week, which we thought would work, and then we did 1 hour twice per week. So Monday, Tuesday, so 2 days in a row. So we took that 6 mg per kg and we split it and gave 3 mg per kg on Monday and 3 mg per kg on Tuesday. And that was predicted to actually, based on the modeling, be the most effective way of keeping HSP90 expression down. And then we split it on a Thursday-Monday schedule, and we did a bunch of biopsies. And the data, the prediction proved to be true. So in the dogs that got the 8-hour infusion, they actually had the best responses to therapy. The ones that had the 1 hour once per week, some of them had some reduction, some of them had mixed responses. The 1 hour twice per week didn't do as well, but the dogs that got the 2-day in a row infusion did really well in terms of clinical responses. But here was the data that was important. If you do the 1 hour infusion once per week, you can see that you get a little bit of knockdown of KIT, but it's really back up within 72 hours, and in this case, it really didn't go away at all. And then if you look at the dogs that got it 2 days in a row, you completely lost KIT expression. So you were able to hold HSP90 expression down. So we had data that showed that the regimen was really critical for this drug, that you had to give that drug 2 days in a row. We also showed that you didn't have to be dose intense about it. You could take the dose that you were supposed to get over the week and split it in half and still get that effect. And so what that did is to mitigate any side effects. And this is an example of one of those dogs that had really a horrendous mast cell tumor that had failed therapy and had this 2-day in a row treatment, and he had a couple cycles of it 2 days in a row, and then had a complete response to therapy, and that tumor actually never came back. And that dog went on to live for several more years. So we were able to predict, using PK modeling, what regimen was most effective. And we were able to show that that was the case using PD analysis. But the 2-day-in-a-row regimen was not evaluated in people because essentially the train had already left the station for the phase 2 and phase 3 studies. So they had already designed them. They had already decided to do once per week. And it ultimately failed in phase 3 studies due to lack of efficacy, which was, I mean, it's disappointing to see that happen. But I think there was such a lag in the initial work we did and then the second study that we did that it was too late by the time we gave them the data. They'd already planned those studies, which is unfortunate because there's an active drug that probably had activity in several tumor settings and it's now not been moved forward. So to summarize, I strongly believe that spontaneous cancer in dogs can be used to explore safety and activity of new therapeutic approaches. Everyone benefits from this approach because we have our pets that gain access to really novel state-of-the-art therapy that they wouldn't have access to, and generally these studies are all free, and then we generate critical information that's useful in the drug development process. And then if we can integrate clinical trials between human and veterinary oncology, we can probably improve the success of translational outcomes. And what I'd like to add at the end is that this effort isn't just restricted to cancer. There's a number of other efforts in other diseases where we're trying to integrate our medicine approaches. So for example, dogs get a disease that's very similar to ALS in people. It's called degenerative myelopathy. A very small percentage of people have a mutation in a gene called SOD1 that drives that disease. And as you know, ALS is a universally fatal disease, so there's no therapeutic that actually works. Same case for the dogs with degenerative myelopathy, but dogs, about 80% of them carry the SOD1 mutation. So dogs are a great model for SOD1 mutant ALS. And in fact, there's a couple of clinical trials that are ongoing right now using an AAV9 vector, other AAV vectors that are carrying a SOD1 short hairpin RNA to knock down SOD1 expression in the CNS. So dogs with this disease are now being leveraged to help develop new therapeutics for that human disease. And with that, I'll stop and say that nothing gets done without help. And there have been a whole slew of people that I've worked with over this course of many of these studies that have been part of the team effort in getting this translational effort done. And now I'll take any questions.
So any questions for Dr. London? That was incredible.
Thanks. It's all so clear. Yeah.
Faster than me. My question is a little off base. So I do spinal cord injury research, and we're considered kind of an orphan standing because there's so few, and drug companies don't invest a lot of money in. I'm wondering from the dog perspective, you have a drug that was effective in dogs. Are there private companies or donors that would rescue that drug? That we heard some of that this morning, that would marshal that for dog owners?
So the question is, are there companies that are picking up some of these leftover drugs and bringing them to the canine market? The
Right.
answer is yes. Okay. There are several smaller companies that are trying to get into that market right now. It's a tough market because we're a cash for service business, so people have to be able to afford whatever therapy is being marketed to them. And so, but there are, many of the companies I've worked with have actually given me the backup compound to work with, not the primary compound. And in 3 of those cases, the backup compound has gone on to approval. So yes, those things do often get moved forward.
So when you say approval, you mean FDA approval?
It's the center, through the Center of Veterinary Medicine.
Okay, but it doesn't restart
The human effort?
the human
No.
effort? No?
No.
Okay.
It's unfortunate.
And there's no way for that to happen?
Well, efforts can get restarted through investigator-initiated efforts. So, I mean, we had talked about with the HSP90 inhibitor potentially getting funding to go back and do a 2-day-in-a-row dosing schedule in people. But it sort of died with the company dying.
So I'm just curious again on the regulations of your dog trials that you've been able to do, the lack of requirements you have, USDA requirements?
So, right, so
Just curious how that works, and that seems really fluid.
Right, it is really fluid. So there is no standard for approval of clinical trials in client-owned animals, and it's a relatively recent phenomenon. In general, IACUC guides or oversees the use of animals in research. And the reason why there's IACUC is that laboratory animals don't have a guardian to make decisions for them. So there's this body that makes decisions. The involvement of client-owned dogs in sort of translational research is relatively new, probably in the last 10 to 20 years. The USDA guidelines are extraordinarily vague, and now the USDA has officially sort of stepped away from it and said, we don't want to be involved in client-owned animal work. So we're all in a vacuum right now. Every institution has sort of a different approval process. Many of us now have a hospital board that approves, and then we also get IACUC approval. But there is now a concerted effort associated with something called the CTSA One Health Alliance, which is an alliance of 12 academic veterinary centers to put a veterinary IRB together so that will form our own structure for approving these clinical trials. And within academic centers, there's at least some structure, but many studies are done now in private practices, and there's absolutely no oversight associated with that. So I think our goal is to create a structure rather than waiting for someone to create one for us.
That's helpful, and you told a story where the FDA definitely valued some of the data you generated. Do you ever see any other issues with, since they're not quarantined in these areas, having concomitant other illnesses? Have you experienced any issues with the drug trials you've been doing in your clinic and how they may be exposed to other elements? Or that's just a natural thing within human studies, but just trying to, when you're doing early development, trying to control that?
So for most of the stuff that we use, there are small molecule inhibitors, and so they're relatively safe. Nearly all of them are excreted in the feces. And the same sort of handling would apply to anything. I mean, you wear gloves sort of thing. But now that we've started to do more viral vector studies, it's become a little bit of an issue. It's not really clear what the guidelines are, and every institution has their own regulation associated with viral vector studies. We have these AAV studies going on for degenerative myelopathy in dogs. We have a couple of immunotherapeutic platforms that are using viral-based delivery to tumors, and there's actually one ongoing now, and they do require some level of quarantine. So they're much more strict for client-owned animals with respect to exposure than they are with humans who go home. Yeah.
Great, thank you.
Okay. Thank you.